target a gene and then trick the cell into repairing the sequence of DNA in that region to something else that might be actually the normal sequence, right? So if someone has, for example, sickle cell anemia is a famous one because it's
Yeah.
got one sequence, one base pair different. You can actually revert back to that sequence and essentially change the cell's sequence of DNA in order to encode for a wild type normal copy of the gene that's encoding in these blood cells that would enable you to transport oxygen better. And so, the sickle cell is not as shriveled up anymore. It's back to its normal state. And that's powerful. And so you can do all kinds of things with this type of technology now. You can engineer drought-resistant crops. You can actually do something called gene drive, where you can affect mosquito populations that are carrying malaria. You can do you can use this in specific ways to use it for immunotherapy and cell gene therapy and cancer. And so it's a broadly applicable technology that allows you to manipulate DNA in creative ways in many different organisms, not just in a dish anymore, but in full-blown organisms from all the way to a human.
I love it. Oh my gosh. So I just want to kind of break this down just to make sure that I'm understanding what you're saying here a little bit.
Sure.
So you're telling me that, like, using like the sickle cell anemia example, right? So if somebody has sickle cell anemia, you're saying that they have, just I mean, it's a very small sequence. You said there's a couple of sequences different from like the proper way it should have been, right, in their DNA. So they take this treatment of CRISPR-Cas9, however they take it and that will alter their DNA so that they are actually a healthy person again. Is that
Yeah, so in
understanding
the
that?
case of sickle cell, the concept is simple because it's carried in your blood and it's a single base paired change, small sequence change. There's less risk that you're going to introduce something that's very problematic because it's large. And so, what I will say as an ethical scientist is, we're still working out all of the kinks on how to do this thing at large scale and inside of
Right.
the side of the body. In the case of sickle cell, you can actually take out someone's blood cells and engineer them ex vivo outside the cell, not outside the cell, but outside the human.
Yeah.
And then you can take the cells in the dish that have been engineered and properly sequenced and confirmed to be now what you would desirably want them to be. And then you can transfuse back into the patient. And because these are the patient's cells themselves, you're able to actually engineer this so that you're not causing the immune system to be inflamed. And this set of cells that's just been introduced back into the system, they can populate and they can go through mitosis, things like that. And so you have the ability to consistently treat a disease that would otherwise be genetically encoded with a engineering outside of the body to go back in. And that's a lot less risky than just to engineer something where you're putting CRISPR directly in. Because, things are still being worked out in terms of the efficiency. And that's, if you were listening to Jennifer or Emmanuel or anyone on that, in that sphere still, there's a very, very careful call to ethics. And, granted, I'm not the person who's always doing the editing myself. My company and my focus is on helping folks get the right tools in place in order to do this. And so if I've misspoken on any of that, I'm sure there's probably new ways to do things that are always evolving. And, ultimately it's just trying to bridge the gap between the folks that are out there in the world that want to hear about this and the folks that are in that space doing it. And it's important to note that when I say there's lacks of efficiencies and things like that, that's why we're not doing germline editing. That's illegal. Worldwide stoppage on that. No one's doing anything like that. These are cures for diseases or for pathology that's associated and attributed to somatic tissue, non-germline, non-reproductive. So we're not changing the inheritance. You're not inheriting designer genes. You're not doing this in a dish with human embryos. So it's important to note that a lot of folks talk about the ethics behind this, and it's premature in my opinion. I think it's important for people to understand that even if we could do that, should we? That's a question, but we're not there yet.
Yeah. Yeah. Which, I mean, I think it's hugely important because, I mean, this is, like you were saying, that's why like the bacterium piece is so critical because, I mean, that could get, it could get dangerous pretty quickly depending on whatever people are doing. But I mean, I think it's important to just for people to be somewhat aware of this. Cause I mean, is it safe to say that CRISPR to some degree is kind of the next thing, right? Like for the next 100 years, like this is what we're going to be, experimenting with, figuring out how to do, like I said, more efficient or just in different ways. I mean, is that safe to say?
Yeah, I mean, Vertex Pharmaceuticals, I believe, is the first one that just had an FDA approval for a CRISPR genome edited focused therapy. So There is an FDA approval already that was just happened. That just happened within the last year. There is no alternative way in order for us to go forward with genetic engineering that's more efficient or better than CRISPR, although we have still a lot to work out. And it's also important to note that it's not just Cas9. There are all these other CRISPR-Cas systems. There's Cas12, 13B, 9, of course, different versions of 9. There are several different ways that people have engineered this for efficiencies and to safeguard against off-target effects from this, things like that, right? And so it's going to be here to stay. And in this type of technology, and the concept of humans using this for enabling the human condition to be improved is not going anywhere. And that's because of a lot of our therapies from the past are imperfect. Not that CRISPR is perfect, but it's more perfect because, when you use a chemotherapeutic cocktail, it's like essentially saying in some cases that you've got one tree in the forest that's not so good and you want to get rid of it, but you're going to light the whole forest on fire to do that. And so we want to become more perfect, more perfect and more precise in our ability to engineer therapies that are for humans, and then do that in a more personalized way, because not all folks have the same genetic backgrounds, and as just as unique as everyone's genetic background is, so is cancer sometimes in many cases. And so, in order for us to be able to better treat folks collectively, we need to become more precise.
Yeah, 100%. Well, and I'm hoping that you'd be willing to speak a little bit more to the ethical piece of this, because I think because like I said, CRISPR and just in general, I think it's just kind of a crazy concept a little bit. And I think the ethical piece is really critical because, well, if you'll forgive me for quoting Jurassic Park, sometimes I think you might know the line I'm going to say here, but sometimes we worry more about if we, what is it? If we could, if we should.
Yeah.
So, I mean, can you just explain?
Had it right.
Yeah. A little more of the ethical piece of this, like what does that look like right now, and like where do you think it's going to go?
Well, I believe that no one has better articulated this than Jennifer herself. I think that it's very important to look and turn around and say, in the face of all this fame and the excitement about CRISPR, Emmanuel and Jennifer, especially because I was at Berkeley a lot during all this, she's been very outspoken about making sure that the technology is used in an ethical way.
Yeah.
I just said that there was a haltage or a no-fly zone for germ editing and germline editing in humans, and that's because that is an ethical thing, because you're changing the course of history by changing someone's genetics, which will be with them forever if you pass those down.
Yep.
Ultimately, there's other ethical parts to this too. If you're talking about something like gene drive, where you can effectively change the mosquito populations in certain areas in the world, or that can affect ecosystems, and that can cause all sorts of collapse, and you have to be very careful about that. I think the answers to these questions and the answers to all of this is that scientists must always continue to communicate every bit they can to everyone, because as it comes up, we learn more and more every year, and it's becoming more and more complicated. And because it's complex, and because we don't understand all the answers all the time, not even close, it's going to become even worse for folks that aren't in that space all the time. I'm certain that I haven't looked at all the literature that's been out there recently about new improvements to these types of technologies or the new therapies that are coming out. How could I? Anyone couldn't do that all day, every day. And so the public that's not in science, it's not that folks are not able to grasp this. It's that the scientists need to be able to continue to communicate complicated things in a simple way. But it's understood as to why that's happening, because scientists aren't the only ones that can have control over the ethics of this. We all need to have continuous conversations about this.